To my YouTube channel. I am Neelima Priyadarshnee Panda, PGT Biology. Today we are going to read about microsporogenesis in plant. Microsporogenesis means production of male gamete in plants. Before reading about microsporogenesis, let us understand the male sex organ present in plants. The male sex organ present in plant is stamen. Stamen is the male sex organ present in plant. The stamen consists of two parts. The stamen consists of two parts. The upper part is called as anther. The long, slender and the stalk is called as filament. The stamen consists of two parts. The upper part is called as anther and the long, slender, the stalk is called as filament. The anther is bilobed and each lobe consists of two theca. Anther is bilobed and each lobe consists of two theca. So let us understand what is theca and what is the structure of anther. The structure of anther is actually it is like this. The structure of anther is like this. Here this is one lobe, this is another lobe and each lobe consists of two theca. So this is one theca, this is another theca, here one more theca and here one more theca. So anther is bilobe, this is one lobe, this is another lobe. Each lobe consists of two theca. So total four theca are present. Inside that some round like structures are present like this. These are some round like structure present inside each theca. That round like structure is nothing but microsporangium. So total four theca are there and each theca one microsporangium is present. So total four microsporangia are present. Inside that microsporangium, a group of cells are present that is called as sporogenous tissue. A group of cells are present that is called as sporogenous tissue. About this sporogenous tissue, we will read later on. The immediate layer of that microsporangium, this is the immediate layer of microsporangium that is called as tapetum. As it is the immediate layer of the microsporangium, so its function is to provide nourishment because it is the immediate layer of the microsporangium. Now, the outer layer of that microsporangium is like this. You can all see here. The outer layer is called as epidermis. In plant, in animal, the outer layer is called as epidermis. It is very easy to remember. In plants and animals, we are going to read the same word, epidermis. Next to that epidermis, the layer is called as endothecium. Next to epidermis, the layer is called as endothecium. Next to that endothecium, the layer is called as, this layer is called as middle layer. And as I have already told that the immediate layer of that microsporangium, is called as tapetum. So total 4 layers are present in a microsporangium. The outer layer is called epidermis. Next to epidermis, the layer is called endothecium. Next to endothecium, the layer is called middle layer. And the immediate layer of microsporangium is called as tapetum. As I told you earlier, the function of tapetum is to provide nourishment to the sporogenous tissue which is present inside that microsporangium. Now come to the function of the first 3 layers. The first 3 layers, epidermis, endothecium and middle layer. Always the outer layer function is to provide protection. Always we know that the outer layer always helps in protection. And it also helps the anther to dehiscence and release of pollen grain. So now once again, the anther is bilobe. Each lobe consists of 2 theca. So total 4 theca are there. Inside that theca, some round-like structure is present that is called as microsporangium. So total 4 theca. So 4 microsporangium are present. Inside that microsporangium, the group of cells are present that is called as sporogenous tissue. About the sporogenous tissue we will read later on. The immediate layer of that microsporangium is called as tapetum. The function of tapetum is to provide nourishment. The outer layer of microsporangium is called as epidermis. Next to epidermis, the layer is called endothecium. Next to endothecium, the layer is called middle layer. And the immediate layer of microsporangium is called tapetum. And the outer 3 layers, epidermis, endothecium and middle layer, the function is to provide protection and help in dehiscence of anther to release pollen grain. Now as I told you, inside the microsporangium, the group of cells are present that is called sporogenous tissue. So sporogenous tissue, now we are going to read. Sporogenous tissue, the function is to provide, later on they will give rise to male gamete. And when the microsporangium further develop, they develop to form pollen sac. So total 4 pollen sac inside which pollen grains are produced. And the pollen grains are going to produce from this sporogenous tissue. So let us understand about microsporogenesis, that is production of male gamete. Microsporogenesis means production of microspore. Microsporangium when further develop, they form pollen sac inside which pollen grains are produced. So as I told you earlier, inside the microsporangium, the group of cells are present. These group of cells are nothing but they are called as sporogenous tissue. The group of cells are called as sporogenous tissue, and this is our microsporangium. Each cell of the sporogenous tissue are called as MMC, that is microspore mother cell. Microspore mother cell. From the name itself, mother, they will give rise to later on the pollen grain, but because mother will generate. So each cell of the sporogenous tissue is microspore mother cell. The microspore mother cell undergoes meiosis, which include meiosis 1 and 2 to form 4 cells. They are called as microspore tetrad. As 4 cells are there, so they are called as microspore tetrad. and these cells are haploid in nature. And microspore mother cell, mother, so they are diploid in nature. Inside the microsporangium, sporogenous tissue is present. Sporogenous tissue consists of group of cells, and each cell is a potential MMC, microspore mother cell. Microspore mother cell will undergo meiosis, which include meiosis 1 as well as 2, and form 4 cells. Those 4 cells are called as microspore tetrad, and they are haploid in nature. And when the anther mature and develop, the microspore dissociate from each other to form pollen grain. When the anther mature and they become dehydrate, then the microspore dissociate from each other to form pollen grain. What happens to these microspores? Let us understand this. The microspore undergoes mitosis to form 2 cells. The larger one is called as vegetative cell. Smaller one is called as generative cell. The larger one is called as vegetative cell, which has abundant food reserve, and the smaller one is called as generative cell. And the generative cell floats in the cytoplasm of vegetative cell. At this stage, it is called as 2-celled stage because it consists of one larger vegetative cell and the smaller generative cell. So 60% of the angiosperm, they are shed at 2-cell stage, vegetative and one generative cell. But about that 40% of the angiosperm, they are shed at 3-cell stage. As for the name itself, answer is there. The generative cell, it will generate. What it will generate? It will generate the male gamete. So the generative cell undergoes mitosis to form 2 male gametes. The generative cell undergoes mitosis to form 2 male gametes, and here one vegetative cell. So 2 male gametes plus 1 vegetative cell. So this is called as 3-celled stage. So 40% of the angiosperm, they are shed at 3-celled stage. 2 male gametes, which are actually given by those generative cell which has undergoes mitosis. And 60% of the angiosperm, they are shed at 2-celled stage. One, the larger one vegetative and the smaller one generative cell. Now, when the anther mature and dehydrate, the microspore dissociate from each other to form pollen grain. So let us understand the structure of pollen grain. The structure of pollen grain. The pollen grain structure is just like this. Here we can see the outer covering of the pollen grain. We can see the outer covering of pollen grain. The pollen grain outer covering is nothing but that is called as exine. The inner covering of pollen grain is called as intine. And inside that the larger cell is called as vegetative cell. The smaller one is called as generative cell. As we can see from the diagram, the intine is continuous. We can see the intine is continuous. But we can see the exine is not continuous. We can see here the exine is not continuous, and that places are called as germ pore. Now the composition of intine is cellulose and pectin, and the exine composition is, they consist a very important component, that is your sporopollenin. Sporopollenin, it is the composition of exine. Sporopollenin can withstand high temperature, as well as they are very much resistant to acid, alkali, enzymatic action, and due to presence of sporopollenin, pollen grain can be preserved as fossil. So the outer covering is exine. The exine is discontinuous at certain places, that is called germ pore, where sporopollenins are absent. The intine, the composition of intine is cellulose and pectin, and inside which vegetative cell and generative cells are present. So this was all about microsporogenesis, that is production of male gamete, structure of pollen grain. Thank you all.